Good afternoon or good morning, depending on where you are, and welcome to the next webinar from HORIBA. And today we're pleased to have really speakers from ColorCon and Innopharma to talk about best practices for multi-particulate formulations and really the importance of computational design and process analytical technology. The speakers today are Chuck Vesey and Chris Callahan. Chris is the head of engineering at Innopharma with a number of years of pharmaceutical analysis and particle analysis experience. And Chuck is formulation technology manager with ColorCon. Again, he's been doing formulations for decades, a lot of experience with coatings. So we're really pleased to have these two speakers with us today. So with that, go ahead, Chuck, if you want to share your screen and take it away.
All right, fantastic. Well, thank you, Jeff, and thank you, HORIBA, for organizing this webinar. Modulating drug release characteristics has traditionally focused on coating thickness to achieve a target release profile. Typically, the quantity of the coating is determined by increasing the percent weight gain until a desired release profile is obtained. This often is a very time-consuming process that does not offer the process control necessary to satisfy increasingly important quality initiatives. In this presentation, Chris O'Callahan and I will discuss computational design tools, process analytical technology, or PAT, and a best practice approach that can help design more robust formulations, predict performance, and accelerate development by reducing traditional iterative trial and testing practices. So as Jeff mentioned, my name is Chuck Vesey. I'm a formulation technology manager here at ColorCon. And for this portion of the webinar, I'll share best practices for formulating multi-particulates that speed development and build quality into these dosage forms. I'll also share a case study tool, namely dynamic image analysis, that was used in a case study used to demonstrate these goals. Okay, the intended outcomes from this webinar include an understanding of how best practices and the technology presented here can accelerate development of multi-particulate products, to demonstrate how computational design tools and PAT support robust formulation development, and how a structured set of best practices can reduce formulation and process risk, not only throughout development, but also throughout the product's life cycle. Before we get started, let's briefly review the benefits of multi-particulates as an approach to oral controlled release. Multi-particulates offer formulation flexibility, meaning different release profiles such as immediate release, delayed release, and sustained release seen here are achievable from a single dosage form simply by changing the properties of the coating. A range of dosing strengths are achievable with a single formulation and manufacturing process. A range of dosage types are achievable with multi-particulates such as capsules, tablets, sachet, and sprinkle dosage forms. And combinations of incompatible drugs are possible with multi-particulates, where this would not be possible with other dosage forms, such as a tablet. Multi-particulates also offer advantages that may include uniform drug distribution throughout the GI tract, the ability to target specific release sites within the GI tract for enhanced local action or systemic absorption, reduced likelihood of dose dumping, and reduced toxicity or GI irritation. These formulation and performance benefits offer us opportunities for improved patient safety, compliance, and therapy outcomes. Now, it's our experience here at ColorCon that a structured best practice approach for multi-particulate success can be grouped into 4 different categories that you see here. These categories include substrate, equipment, coating, and process. And as you'll notice, each category has multiple subcategory attributes to be considered for success also. For example, under substrate, not only do we consider the starting core, we also consider API morphology and binder selection when drug layering. Under coating, for example, we consider things like film properties critical to efficient film formation. We also consider the influence of coating viscosity and nozzle performance on droplet size, and the potential for particle agglomeration during coating. Now, while we won't be able to cover this entire list here today, we'll briefly cover one topic from each area, starting with, of course, the core and substrate selection. Understanding factors influencing release from a multiparticulate under coating, and computational design and process monitoring techniques under the process and equipment areas. So let's start at the beginning, or in this case, the core, where the quality of a multiparticulate formulation and robust performance starts. If we look at this graphic here on this slide, we can see how a uniform consistency of coating layers depends on the consistency of the starting core. Starting with the core, a drug layer is added, followed by a seal coating, a functional coating layer, and lastly, an optional clear or pigmented protective coating layer over the functional coating. The uniformity of these coating layers starts with the uniformity of the core. A uniform size and shape of the starting core allows a more uniform application of each subsequent coating layer. This is why we take an inside-out approach to consistency. So what are the quality attributes of a starting core material that should be considered critical to success? Let's consider particle size distribution, sphericity, and friability. Particle size distribution is perhaps the most important quality attribute of your starting material, whether it's a sugar, MCC, or mannitol sphere, or an extruded spheronized bead, or an active API particle. Changes in batch-to-batch particle size distribution contributes to changes in batch surface area, impacting controlled release coating layer thickness and potentially impacting dissolution performance. I'll mention the concept of particle size distribution and surface area throughout the presentation, stressing its importance to performance. Sphericity. Changes in sphericity or morphology of the starting core material will result in changes, again, in total surface area from batch to batch. Again, this can potentially impact film thickness and dissolution performance. Changes in sphericity can also impact material flow within the fluid bed product chamber, particularly in the down bed direction. Decreases in sphericity may impact packing density and flow, causing sluggish bed movement, dead spots, and ultimately changes in up bed density, which can lead to low coating process efficiency. Friability. Friability of the uncoated core at early stages of the drug layering process will cause changes in the total surface area of the batch. And if serious enough, this will result in changes in film thickness of any subsequently applied controlled release coating layer, leading to changes in dissolution performance. So the take home here and our best practice approach would suggest working with a supplier who understands and controls these quality attributes as part of, per se, a specification, and designing a fluid bed coating process that avoids attrition and friability during processing. To better understand formulation performance, we need to understand what controls drug release from a multiparticulate. Drug release occurs via diffusion to a semi-permeable membrane. The rate of drug diffusion is primarily controlled by two mechanisms. One, modulating film thickness or diffusional path length, denoted as H in Fick's law, and by altering the permeability of the coating with the addition of permeability enhancers, often referred to as pore formers. We can see the impact of increasing film thickness on drug release from a multiparticulate in the graph here to the right. Increases in film thickness result in a decrease in drug release, measured here at a specific time point. However, if we are only considering the effect of film thickness on drug release, we are not seeing the whole picture. Take a look at Fick's law again. We need to also consider the effective surface area. Here we can see the relationship of surface area to film thickness ratio and its impact on dissolution. Graphing data in this way provides us with valuable information about the sensitivity of our formulations to changes in surface area and film thickness. For example, the graph may tell us areas along the x-axis where changes in surface area to film thickness ratio may result in large or unacceptable changes in dissolution performance, suggesting sensitivity of the formulation and areas along the x-axis we may want to avoid. So the take home here is taking surface area to film thickness ratio into consideration, we can begin to have a greater understanding of the impact of surface area on batch-to-batch dissolution performance. Like the previous slide, if we were only considering the effect of film thickness on drug release, we were not seeing the whole picture. Take a look at Fick's law again. We also need to consider the effect of permeability or diffusivity of the coating being applied. We see this in the graph here to the right, where the addition of a more hydrophilic polymer to the semi-permeable coating increased the apparent permeability of the coating across a wide range of film thicknesses from 2 to 20% weight gain. We can see a similar effect when we examine the ratio of a hydrophobic to hydrophilic polymers on diffusion coefficient. Not surprisingly, as the percentage of hydrophilic polymers increase, diffusivity increases. Also of note here, variability in diffusion coefficient appears to increase as the percentage of hydrophilic polymer increases above a concentration of 10%. Now that we understand the importance of the starting substrate and the levers that actually control drug release, let's consider some tools and technologies for multiparticulate design and processing success. Best practice recommendations include the use of computational tools such as MyDosage Design from ColorCon to aid in the design of a multiparticulate prior to going into the lab. Once in the lab, the use of at-line or in-line particle size measurement capabilities to track particle growth is highly recommended and would be considered a best practice. Our experience at ColorCon includes the use of dynamic image analysis for particle size and shape measurements using at-line and in-line capabilities. Let's consider the use of the MyDosage Design tool to help construct a multiparticulate dosage form. MyDosage Design is a theoretical modeling tool that helps determine the optimal starting particle size, coating coverage, volume of the final dose, and capsule size requirements. By calculating these product characteristics prior to initiating laboratory trials, we can avoid time-consuming and costly experimentation. Input functions to the design tool include starting substrate size, desired final dose, and coating layer information. Output calculations include intermediate and finished product particle size, film thickness information, coating coverage, assay, and capsule fill volumes. Now, the great thing about the MyDosage Design tool is that it allows comparison of various design simulations side by side to determine the impact of substrate and formulation changes on these output values. So the next two slides contain particle size data using both at-line and in-line dynamic image analysis capabilities. In the graph here to the left, calculated and measured particle size data collected at-line is plotted versus theoretical weight gain. The observed trend shows consistent particle growth between sampling points versus calculated values. The data trend also illustrates the value of dynamic image analysis for fluid bed process tracking. If we then plot particle size versus dissolution data, we can see a strong correlation between particle size or film thickness and dissolution performance. By combining predicted values from our computational design tool and in-process tracking enables a greater understanding of the relationship between film thickness and dissolution performance. So at this time, I'd like to hand the virtual podium to our co-presenter today, Chris O'Callaghan.
Okay, so thank you, Chuck, and thanks to HORIBA for arranging all of this for us. So I am Chris O'Callaghan from Innopharma Technology. I'm head of engineering here at Innopharma Technology, and if you're trying to place the accent, Ireland, Dublin in Ireland is where we're based. In this brief section, I'll only take about 10 minutes. I'm just going to talk a little about Innopharma and the products that we have, about the importance of process analytical technology, which I think that Chuck has set me up for very nicely, and then about the ICON 2 itself, which is the technology used in the case study Chuck is going to come back and present just after my section. So I'll talk about applications, tech specs, and a little about the method of operation itself of the technology. Innopharma Technology, first of all, was founded in 2009 by Ian Jones, our CEO. And there are 3 divisions to the company. So beyond ourselves in the technology part of the company, there's also an education division where we focus on upskilling people to work within the pharma and medical device industries, and a technical contracting services division focusing on validation engineers and laboratory QC analysts. And across those 3 divisions of the company, we have about 75 employees currently. The image in the bottom right of the slide there is from our 10th birthday as a company last year, back when we were all allowed to gather together in big groups and take photographs. Anyway, we don't have to, we see enough of that stuff in the news. Our products, first one here is the ICON 2. That's a direct imaging particle analyzer. I'll be talking about that in more detail in a couple of slides, but in addition to that, we also have a multi-point, up to 4-channel NIR spectrophotometer called the Multi-I2, and we have a system called SmartX, which is described here as a vertically integrated platform for smart process development and manufacturing, and is really a logical evolution, we think, to being a PAT developer. Where we've built a system now to help enable customers make better use of the data generated by PAT in their facilities and really feed that forward into more advanced process control methodologies and approaches. Where we've been over the last 11 years kind of speaks to this evolutionary process as well. We started out in 2009 with the first generation ICON, and the first generation Multi-I followed soon after. These then became the ICON 2 and the Multi-I 2 based on market feedback, based on learnings and so forth. But in more recent years, starting the development project from 2016, we have been working on this SmartX Industry 4.0 Pharma 4.0 system. Firstly, an application to fluid bed granulation and coating, but laterally then on crystallization, and most recently looking at twin screw granulation and extrusion processes, which is a project that I spend quite a bit of my time on at the moment, and certainly a very interesting process. On the importance of PAT, and Chuck set this up very nicely, talking about critical quality attributes of incoming raw materials. And the reason we focus so much on the critical quality attributes of incoming raw materials is they have the capacity to dramatically impact either how our process runs or the end result material we'll end up with. And the traditional view is to characterize your incoming raw material critical quality attributes, and then provided they fall within a certain acceptable range, traditionally, perhaps we run our process as per its standard definition. But a lot of this comes from the lack of tools to measure these attributes in real time in processing. And that's where PAT comes in, with the ability to measure things like particle size in line in real time and moisture content as well. So the ability to monitor these, not just at the start of the process, but throughout, lets us adapt our process inputs rather than keeping them as rigid. We can adapt them to ensure the best possible critical quality attributes of our finished materials. So really, PAT helps us understand processes. Whether we're adapting these parameters in real time with an advanced control system, or whether we're looking at this as an opportunity for longer term process control improvements, really comes down to the type of process, the type of industry. And with a bit of a pharma focus in this presentation, of course, yes, making changes to a process on the fly is not particularly kosher, but process improvement projects are very, very common, which take a slightly longer term view approach to the same thing. So better process understanding ultimately leads to more robust and reliable processes, better yields, less waste, less cost. And this is something that PAT and of course selection of good quality, reliable raw materials all play into. So on to the Icon 2 itself. I've already covered that it's a real-time particle sized distribution and shape measurement device with applications in R&D, scale-up, tech transfer, and in both batch and continuous manufacturing. And then some of the uses are listed there: fluid bed, coating, granulation, drying, twin screw, roller compaction, extrusion, spheronization, and several others also. On the left-hand side, you can see an image of just the bare Icon system up the top, and in the bottom left, we can see it in its at-line configuration. So this device is usable both in-line, measuring through a window on a process, and in an at-line setting with this automated benchtop stage, which moves the sample material around below the camera system, allowing it to take multiple images and analyze the product. And then over the right-hand side, you can see a couple of example images of some multi-particulates and of some fairly rough-looking granulate, which I can't quite recall the provenance of. It could be twin screw, maybe. The Icon is connected up to our Ipass software, particle analysis software system is what the PASS stands for. This is just a screenshot of the real-time software, and then on the right-hand side, a PDF report generated by the system to summarize a batch or a measurement run. And I won't go into any great detail here, but on the left, you can see the real-time image from the Icon, which is essentially giving you your real-time inline microscopy for free. So we can tell a huge amount from the way the material appears in that image. Subjective, certainly, but very, very useful to a skilled or experienced operator. On the right-hand side, then, we have the particle size data, the dV10, dV50, dV90, with respect to time, and we have numeric particle size distribution data and a histogram S-curve in the bottom right. All essentially different ways of representing the same type of thing. In terms of technical specifications, I'm not going to get into this in any real detail. You can read yourselves here, and the handout attached to this webinar has a lot more detailed information on the Icon 2. The only thing I'd point to is the size range of 50 to 5,500 microns. Means that the Icon 2 is not necessarily applicable for all incoming raw materials, but generally any material where we have begun a process of agglomeration, whether we're granulating it, whether we're extruding it. Typically, at least in kind of pharma processing, puts us well into that range, 1, 2, 3, 4, 500 microns, which is just the butter zone for the system, the kind of ideal range to use it in. So while it may not work on your micronized APIs and your incoming raw material lactose, for example, once you start to granulate that, it's an excellent way of keeping track of your processes and your material performance at that point. The method of operation, it is an imaging system, as we've kind of alluded to already. And in this image here on the left-hand side, you can see the camera lens and you can see a bank of LEDs arranged around that. And these LEDs are arranged in red, green, and blue banks. And they flash to, it's flash imaging, it's not, it probably didn't need an animation to explain. They flash to simply allow us to measure fast moving particles without any smearing effect. The camera frame rate doesn't go quite high enough, so instead we use it at extremely short duration pulse of light, about 5 microseconds long, to allow us to measure particles moving at up to around 10 to 12 meters per second. I mentioned that the LEDs are in banks of red, green, and blue, and this is how we get this interesting distribution of colors on the particles you can see in this image. So this image has not been modified or adapted, it's just a standard image out of the icon, aside from making it round so it looks nice in this presentation. But this color information, in addition to the differences in brightness of the pixels, allows our analysis algorithms to better determine when we see two particles overlapping like this. So the difference in color and intensity allows the algorithm to pick out that this is one particle overlaying another one rather than simply one large particle. And I'm sure you can see the importance of it even here, but you can imagine when you're looking at a process like fluid bed granulation through a window where it's essentially a tightly packed sheet of material on the other side of the window. It's very, very important to be able to tell the difference between small particles all packed together touching one another versus one giant particle or several very large agglomerates. That's image capture. Then once we've captured the images, how do we turn those into particle data? And I have to hold my hands up, my webcam's not on, but I have to hold my hands up at this point and say these image analysis algorithms have been written by people with a lot more understanding than me. So you're going to get the executive summary, partly because that's all we have time for, and partly because that's all I really understand anyway. So we identify the edge pixels of likely particles, and at that point the system fits ellipses to all of these possible particles. And you can see these ellipses demonstrated in the dashed green lines. The image on the left shows that there are several different ways that we can characterize a particle that we take an image of. So our approach is this elliptical fit with the green dashed line, which gives us a major and minor axis measurement. Parameters like ferrite diameter, Martin's diameter are also indicated on here. The reason we make such a big deal about this elliptical fit is that a huge number of the technologies out there use essentially assumed sphericity. Assumed sphericity, try saying that quickly. And you can see that indicated here in the center image, where every particle is modeled based on a single diametric measurement rather than the two shown below in the green ellipses. And this elliptical fit just helps us be a little bit more representative of the true volume of these particles, because it all ultimately boils down to volume, as I'll show in a moment, as well as allowing us a basic understanding of the shape information of these particles also. There's just a very quick graphic there showing an ellipse fitting over a particle, which moved a little bit. I don't think we need to worry about that too much. And then I believe this is my final slide. So before I hand back over to Chuck, I'll just outline what these DV10 or D10, D50, D90 things we keep referring to are, because I'm sure 90% of the audience are well up on this, but just bear with me for a moment. For the few people who may not have come across this method of particle sizing before. So D values are computed based on the ellipses that we fit to individual particles, and they are descriptors of the particle size distribution, but not in maybe as straightforward a way as you might assume it would be done mathematically. First of all, they deal with volume or mass rather than strictly particles by number. And they are percentile measurements. So the easiest one to start with is the D50, shown in the bottom right, which essentially divides the population of particles when arranged in order of smallest to largest volumetrically. It divides it with a 50-50 weight or volume split. So it's the diameter at which half of the population of particles is smaller and half is larger by volume. Similarly, the D10 is just the 10th percentile, the lower end of the distribution, and the D90, not shown here, takes very much the upper end of the distribution. This comes originally from sieve analysis, where we don't have the opportunity to count the number of particles in a sieve, just to measure the mass of particles within that sieve. So that's why this D10, D50, D90 approach is used and has been used for a long time, and more than likely will continue to be used for a long time in particle processing industries. So hopefully that will help you understand the next set of slides that Chuck is going to get into around the case study that we carried out in ColorCon a couple of years back. So with that, I thank you very much for your attention, and I'll hand you back over to Chuck.
All right, fantastic, Chris. Thank you very much. And now for our case study, in which we used the Icon 2 to examine particle size in using dynamic image analysis and a fluidized bed. Okay, so in this case study, a BCS class 1 freely soluble API was layered onto a sugarlet sugar sphere using a GLAT GPC-G2 equipped with a 7-inch Wurster insert. And you can see that here in the picture to the upper right. 2 fully formulated ethyl cellulose-based coating systems, namely Sure-Release and OpenDry EC, were used in the study. The Sure-Release is an aqueous-based pseudo-latex dispersion of ethyl cellulose, whereas the OpenDry EC is a dry dispersion of ethyl cellulose polymer intended for use from organic solvents. For our case study today, I'll only cover the aqueous-based work that was part of the case study. As such, you'll note the process parameters that were used in order to be able to apply the Sure-Release during the coating. Now, our study response variables included in-process particle size analysis by the Icon inline, and we also had at-line capabilities with a cam sizer. We also examined particle morphology, assay, and of course dissolution testing. And from the particle size data, we calculated film thickness and examined the relationship between film thickness and dissolution performance. And as you can see in the picture to the left, we see the familiar Icon 2 collecting data on our GLAT fluid bed here at ColorCon during the case study. Now, as part of the study, we did notice a slight bias in growth of larger particles when the particle diameter was plotted against average growth during the coating process, as seen here in the graph to the left. This bias occurs for several reasons related to substrate flow, specifically in the up-bed direction. Those reasons can include that particles with a larger cross-sectional area will absorb more coating when traveling through the up-bed region of the spray zone. Heavier particles by mass also move slower through the up-bed, increasing dwell time in the spray zone. And larger particles by mass fluidize lower in the up-bed direction, resulting in more passes through the coating zone. It is for these reasons that larger particles can gain more coating over the course of a coating trial. This difference in particle growth can also be noticed with the formation of agglomerates during coatings. Agglomerates result in an overall reduction in burnt batch surface area. Therefore, the amount of coating on all bead populations increase. These differences can also contribute to differences in assay of discrete beads versus the agglomerates. Now, the take home here, as I mentioned earlier, is a starting substrate of narrow particle size distributions will minimize this effect. So if we take a look at some data from the study, In this graph here to the left, particle size data is plotted as measured film thickness versus theoretical weight gain. Here we can see a consistent film thickness growth is observed throughout the coating. The observed trend demonstrates consistent growth between sampling points, indicating a process that is in control with no observed deviations. This is the most basic in-process data that is available via this technology. To a formulation scientist or fluid bed operator, this data is invaluable. Let's agree, trouble isn't always apparent when operating a fluid bed. From our computational design tool, we can calculate film thickness values with increasing theoretical weight gain or coating applied for any substrate or coating. We can then use the inline or at-line measurements to track the coating process to ensure the measured particle growth agrees with calculated values. This approach provides added confidence that the process is in control and the resulting analytical data is correct. Here we see dissolution results from the study. A few things to note. Dissolution curves represent 4 different weight gains of 5, 10, 15, and 20% weight gain, and film thickness values ranging from 5 to 35 microns. Although somewhat difficult to see, 2 additional curves are shown here at 20% weight gain following a post-coating thermal treatment or curing step for 30 and 60 minutes. The curing was done at an elevated temperature above the glass transition temperature of the coating in the fluid bed. From the graph, we do see a steady decrease in release with increasing film thickness. Dissolution results agree with measured particle size growth between sampling points and a steady process trend during coating. It is worth mentioning that although a slight decrease in release was observed for those cured samples, the F2 values or similarity factors for the cured versus the uncured samples was calculated at greater than 60, indicating similarity. Now, instead of looking at the data as percent dissolved versus time, let's group the data by time point and graph percent dissolved versus film thickness. And that's what we see here. Here, percent dissolved data is plotted against film thickness at the 2-hour time point, or 120 minutes. Moving from left to right, we see a decrease in release with increasing film thickness. Additionally, a polynomial fit is added to the data with the equation and the R-squared value. Now, let's look at the additional release data plotted in a similar way. And here we see all the dissolution data plotted by time point versus film thickness. And again, we see similar trends of decreases in release with increasing film thickness, as noted on the previous graph. Lastly, dissolution data from a repeat coating trial is overlaid with the calculated values to demonstrate the potential value of a model-based prediction of release performance. In addition to the potential value to performance predictions, several other benefits of computational design tool and PAT are realized, and these include greater process understanding throughout the development and scale-up studies, availability of real-time particle size and early detection of an out-of-process trend, cost avoidance due to process interruptions, low yields, and possible batch loss, and lastly, the potential use as a fluid bed process control tool in manufacturing. To summarize, multi-particulate dosage forms offer formulation flexibility that ultimately contribute to patient-friendly features such as once-a-day dosing, improved swallowability, and taste masking options for specific patient populations. Robust performance of multi-particulate formulations starts with the core. Building the active layer and functional coating layers on a consistent substrate is key to building quality into multi-particulate formulations. Using computational design tools and dynamic image analysis like the ICON 2 offer the opportunity for enhanced formulation and process insight. And lastly, a best practice approach to multi-particulate development offers opportunities to improve outcomes, speed development, and helps ensure product robustness. I'd like to finish with a quote from one of my fellow fluid bed enthusiasts here at ColorCon. Designing your multi-particulate product and manufacturing process with a set of best practices in mind will expedite the development process and help ensure a trouble-free life cycle. I share this quote with you as it summarizes the value proposition of the concepts that we've presented here. ColorCon's best practices, computational design tools, and a structured process development approach utilizing PAT are keys to successful multi-particulate commercialization and a trouble-free life cycle. As we finish up, I'd like to thank my ColorCon and industry colleagues for helping make this work possible. Piyush Patel, my formulation technology partner here at ColorCon. Ed Godak, process technology at Glatt Air Techniques. Our code presenter today, Chris O'Callaghan, head of engineering at Innopharma Technology, and of course our host here, Jeff Bodycomb, product manager at HORIBA Scientific, and my industry contact for everything particle size and shape related. It's collaboration with industry colleagues like these that share mutual interests that adds value to our work and offers the opportunity to bring forward solutions that solve shared industry challenges. In closing, I hope we've been able to convey the value of a best practice approach to multi-particulate dosage form development. If you would like more information, you can connect with ColorCon via the ColorCon website. We are currently highlighting a multi-part series on fluid bed best practices. Visit the ColorCon website at colorcon.com and look for Connect with ColorCon for more information. Thank you, and at this time, I'll hand the virtual podium back to Jeff for any Q&A.
Okay, well, thank you very much. That was really an interesting presentation. Really appreciate it, both Chuck and Chris. So we're going to start working through some of the questions that have arrived during the presentation, and then feel free to add to the list. So I guess the first question, what is the best techniques for creating multi-particulates and maintaining their integrity during capsule filling?
Okay, I guess I can handle that question, Jeff. Thank you very much. So for me and the experience that we have here, creating a multi-particulate and being able to maintain its integrity, I mean, fluidized bed technologies, whether you're using Wurster, and in some cases, perhaps other approaches such as, top spray or tangential or even powder layering. Fluid bed by far is the best technology to use to create a multi-particulate dosage form. Our recommendations and a best practice that we would make would be to apply that optional top coat or seal coat over your functional coating layer. So applying a clear, perhaps HPMC based top coat. It can be clear, it can be pigmented in certain situations if you choose, and applying that to the outer over the functional coating layer. That way you provide that additional protection to the bead once you've gone through all the trouble of drug layering and applying your modified release coating layers. So for us, it would be the application of a protective coating layer on the outside, and that will certainly be able to protect the bead and your investment from downstream unit operations, such as packaging and encapsulation and packaging.
Great, thank you. I guess this probably gonna go to Chris. How does the icon handle a coating that changes color?
So that can be challenging depending on the range of color change. The icon really works best with light colored materials, and as we find in pharma, the vast majority of what's produced is white anyway, at least until maybe receiving a final colored coat. But broadly speaking, changes within the light color range, kind of light blues, greens, reds, or going from white to one of these colors is okay, provided we don't darken the particles too significantly. But we have certainly seen applications where we've struggled there, and applications where the change in reflectiveness of the particle has been too significant to keep track of it throughout the process with the icon. So really better used on these white, cream, or light colored materials.
Okay, so for multi-particulate processes and how your excipients impact the process, so they're asking, I guess, about the CQAs and your support. That's probably the Chuck question.
Sure. Thanks, Jeff. So I think we covered critical quality attributes, certainly of the substrate. So in the presentation, I believe early on, we talked about particle size distribution, friability, and sphericity. Those would be my top quality, critical quality attributes for a multi-particulate, and specifically the substrate. And that goes for any substrate, whether it be, like I said, a sugar sphere in the case of the work here done today, and the sugarlets, or, a MCC sphere or other type of sphere. So it's really comes down to your starting particle quality attributes that you really want to be concerned about, not only from during development, but each stage of development through scale up, and then throughout the life cycle of the product during commercialization. And other quality attributes, of course, for other ingredients within your formulation. For me personally, I like to keep the formulation fairly simple. The simpler, the better, and to ensure that robustness and consistent performance.
Okay, thanks. Can this technology be used for other industries such as agrochemicals?
I'll take that in relation to the icon too, I suppose. Yes, certainly. Although it does interlink with the previous point around colors, we have worked on several fertilizer applications over the years. And for the most part, aside from extremely dark colored materials that might contain high amounts of ash, for the most part, those are measurable. So they're not, you wouldn't call them light colored. They are kind of, medium browns typically. But where we're not dealing with a significant color change within the formulation, we can make some adaptations to work with these darker colored materials within limits. Being a camera-based technology, if it can't see it clearly, it can't measure its size. So that goes for extremely dark materials that don't reflect enough of the light back. That goes for transparent materials, where the light refracts through in bizarre ways. And it also goes for extremely highly reflective materials like ball bearings, for example. We can't measure a ball bearing because we see a warped reflection of the icon zone illuminating LEDs rather than the bearing itself. But agrochemicals, yes, we've worked on several fertilizer applications, and aside from the kind of the ones that contain large amounts of dark ash and black materials, they have broadly been successful.
Okay. Next one, how much process understanding is needed in early clinical stages?
Okay, I guess that's going to me. So yep. Process understanding, I guess it depends on whether you're talking about the fluidized bed or whether you're talking about the Icon 2. I'm not sure which way to take that, but for the Icon, for dynamic image analysis in general, and certainly the Icon 2 for that matter, not a whole lot. I mean, these pieces of equipment are very user-friendly. I would certainly start using these technologies from early stages of development all the way through to, when you start getting to those pivotal studies and development batches, demonstration batches, and clinical trials materials. Certainly use it through this technology throughout all those phases. If you're talking about fluidized bed technology, of course, it can be complex, but this is the reason why myself and my colleagues here at ColorCon have been working on this set of best practices, and it is quite extensive. We have quite a few best practices that we would recommend, as I mentioned, regarding the core, the coating, of course, the equipment, and the coating process. So we cover many different areas with our best practices to make this process much more accessible and easier for our clients.
Okay, great. So is there I guess this is overchecked, is there a software that's for suspension-based formulations? I know you talked about your solid dosage form software or prediction tool. Is there something for suspensions?
Yes, about that, yes. So if you're using a solid particle substrate that's going into a suspension to start with, say that you're loading your drug onto, yes, you can absolutely use this technology and our computational design tool for that. So if you're loading onto, let's say, some type of substrate, maybe it's an API particle where you're layering on various controlled release coatings, or perhaps it's an ion exchange resin particle where you're layering on a certain amount of coating. Yes, it can be used in that way. Now, of course, the the accuracy improves as, you know, as I mentioned, the critical quality attributes. So the particle size distribution and sphericity. As the sphericity goes up, your accuracy, the computational design tool will improve.
Okay, great. All right, so next one. How much contrast is needed with the background? For example, Chris, can you use the icon with the transparent process chamber?
So good question. And it makes me think about that slide that explains the image analysis, and that maybe the image on that slide where we see some spherical particles falling on a black background might not be the best way to describe what I'm describing. So we don't need any background contrast really for measuring these particles, because where you come back to the example I said, but I didn't show an image of it, of something like fluid bed granulation, there is no contrast because there is no background. We just see a densely packed window of particles all up on top of one another. And in this case, the system has to be able to pick out the edges of particles in amongst other particles. Now, it doesn't have X-ray vision. It can only measure what it sees at the surface. There's no penetration depth per se, but it will depend on the process type, and certainly we wouldn't want an extremely bright light source on the other side of this transparent process chamber. But in reality, even if we had a bright light source there, we wouldn't see it, because if you're looking at something like fluid bed granulation or coating down within the bed region, nothing comes through from the other side. All we see is particles and more particles behind those particles, and so forth. So there's no contrast requirement except for very edge cases.
Okay. This next one looks like one for Chuck. What particle size distribution range is recommended for incoming sugar spheres?
Well, that depends on, of course, what's your end goal with respect to the fluidized bed and your dosage form. So the sugarlets that ColorCon offers, we have multiple different grades that encompass different particle size ranges and distributions. So that's part of what you would use the computational design tool for. So before you even select the substrate size, using the computational design tool to decide which starting substrate is the best for your intended dosage form based on, let's say, your dose and the multiple coating layers that would be added towards that. I would always recommend, of course, that the particle size distribution be as narrow as possible. And not only that, but even more importantly, that particle size distribution is repeatable from batch to batch to batch to batch, or lot to lot to lot in the case of a supplier.
Okay, great. And when coating beads with IR and ER drug layers, what are the most critical parameters to consider?
That's a great question, and it's an extensive question that would take quite a bit more time to go into than we probably have here today. But I would always recommend finding out and determining throughout the development process what are those critical process parameters. I did mention on the one slide what the process parameters were for the sure release coating that we used in the case study. Here at ColorCon, what we do is we try to determine what those critical process parameters are for our clients before we provide a coating solution to them based on their needs. So in the case of the sure release, things like for an aqueous latex dispersion would be your processing temperature, so your product temperature, spray rates, dispersion solids content, and those kinds of things, those not only variables that would go into your process parameters and coating parameters would be determined by ColorCon, and we would make those recommendations to you when using those coatings. Okay, so I would always recommend a DOE type like process throughout the development process to determine what your critical process variables are.
Okay, I'm going to ask my own question real quickly. Can you remind us of how to reach? You said ColorCon.
ColorCon.com.
Yes, .com, and you said something else.
Okay, sure. So what we have at ColorCon right now, we're highlighting on our website, Connect with ColorCon. So you're looking for the Connect with ColorCon banner.
Okay,
and
great.
Under that banner, we have various different topics that you can examine, and we're highlighting right now a multi-part series on fluid bed best practices. So many of the things that we talked about here today and some additional ones will be highlighted there. So colorcon.com and look for the Connect with ColorCon banner.
Great. Okay, what is the smallest coding difference that you can measure with the icon?
This is always a good question. Interestingly, now there are always lots of variables. I feel like I feel like I'm cagey about every answer, but interestingly, we can measure smaller differences in coding thickness than the size of our individual pixels, because we don't just measure a single particle, we measure thousands and thousands of particles. We can be more accurate the more time we take to make a measurement, so the longer our integration time is, and we can be more accurate in smaller product containers, because based on trials I've done at different sizes in Wurster coders, there seems to be more oscillation in the size of particles seen at any one point in the process over time in the larger beds than smaller. And I guess that kind of makes sense. The smaller ones having a lot less material in them homogenize better. What's the answer, Chris? Yeah, and 2 to 4 microns, plus or minus 1 to 2 microns, but that is taking our time to produce that measurement over approximately, 1 to 2 minutes of measurement in a small bowl system, much like the GPCG2 from the case study that we talked about today. So as good as plus or minus 1 to 2 microns, but your mileage may vary depending on material types and other processing variables.
Okay, another one for you, Chris. Is the Icon 2 EXP, I imagine that's explosion proof, can it be used with solvent-based fluid bed applications?
Yes, it is, and yes, it can. It is ATEX Zone 222, so that's for dusts and gases, and then depending on the part of the world you're in, there is an IECEX equivalent version also available.
Okay. Well, I guess Gina is left, but can you comment a little bit on the role of computational modeling in these applications? I'm not sure whether that's which you want to take that.
Can you read that again, Jeff?
Yeah, can you elaborate a little bit on the role of computational modeling and design in these applications?
So I think we did cover that quite a good bit. If there's, if the attendee who asked the question wants to reach out to me specifically, we can certainly talk more about it. But the computational design tool, I think, has been extremely helpful to us here at ColorCon and extremely helpful to clients, because we use it to plan our work before we even go into the lab. So before we even open up a drum of sugar spheres, before we even put the glass together, we use it to run different simulations of perhaps different bead sizes, different coating combinations, in order to determine what the final dose might be, the capsule fill volume, and how much coating we're actually going to have to apply in order to be able to get a certain film thickness. So it's an extremely valuable tool in that way. And we can actually take those simulations into the lab and actually execute on those simulations in order to be able to get an idea of what the release profile is from different simulations. And that in and by itself to us has proven that to us that we can significantly cut down the amount of trial and test iterations that we go through when doing work in the lab. So that's been the real value for the computational design tool. And of course, when you pair that with the Icon 2 or dynamic image analysis, in order to be able to get you that film data, then you can start looking at, well, what's my dissolution performance at various weight gains on in my simulation? And that becomes a very powerful set of tools.
Okay, we're kind of winding up here in terms of time, so if you have any last minute questions, I do have one. How do you calculate coating thickness?
Okay, either Chris or I can handle that. It's simply just a subtraction method. So you're going to look at your initial particle size diameter in the fluidized bed, or either inline or at line using cam sizer or a similar technology. And you're simply just, as you apply coating, it's simply looking at the particle diameter at time 0 versus particle diameter at some point during the coating process. And it's simply a subtraction method.
Okay, great. Well, I think that's about it. And we're kind of out of time. So if you have any further questions or something occurs to you later, feel free to reach out to us at labinfo@horiba.com, or to ColorCon through their website or via me, and I'll talk to Chuck and Chris, and we'll get those questions answered. Thank you, everybody. Bye for now.
Thank you all very much.
Thank you.